another way.
Oh, good.
So tonight, Dr. Gary Bernard is our speaker, and the title of his talk is Red Blindness, Red Vision, and Far Red Vision of Butterflies, and then some fun stuff. Gary is an affiliate professor of electrical engineering at the University of Washington, and he's a longtime WBA member. He's not only a researcher, but also a field worker, notably participating in a 2009 butterfly survey of Mount Tampotika on the Indonesian island of Sulawesi. Gary started academic life as a 9-year student of electrical engineering at the University of Washington, working on radiated and guided electromagnetic waves. Then he moved to MIT in 1965 to teach that subject. After a year there, he was contacted by a Yale physiologist, of all things, Bill Miller, who was doing electron microscopy to understand dark adaptation of moth eyes and changes in eyeshine intensity. Bill loaned Gary a photo microscope and a net, and Gary proceeded to study everything he caught, including a satyr butterfly. It had a tiny eyeshine spot that was either red or green, but he couldn't photograph it because the spot quickly disappeared after the light was turned on. So, long story short, Gary moved to Yale, where he built computer controlled optical apparatuses to measure eyeshine. In 1976, traveling to Tucson for a meeting, he caught a number of butterflies, including Apodemia mexicanus. I'm going to guess on pronunciation on that. Measuring the spectral properties of eyeshine decay, Gary learned that some species could not see red in the red part of the electromagnetic spectrum, while others could, especially Apodemia. He wrote a paper and submitted it to Nature, but it was rejected by the reviewer who wrote, everybody knows that insects can see UV but not red, and birds can see red but not UV. Gary not only took that as a challenge, but he has proved that some butterflies certainly can see red very well, as you shall soon learn in his talk. So thank you so much for being with us tonight, Gary. So the floor is yours.
Oh, thank you, Paulette. Okay. Yeah, vision, invertebrate vision. It's a really difficult subject and absolutely demands multidisciplinary teaming. These are my collaborators over the years, a lot of them. My talk is about butterfly vision, but it is not my work, it's our work. All of these people are involved. I'd like to tell you a bit about each person, but don't have time. So for this talk, I'll only tell you about the several particularly important contributors to what I'm showing you tonight. Naomi Pierce was a Yale undergraduate who took the basic course taught by world-class entomologist Charles Remington. I gave lectures on invertebrate vision to his class that included my work on butterfly eyeshine. Nomi liked it. In 1991, Nomi established the Pierce Lab in the Harvard Department of Organismal and Evolutionary Biology. In 1995, Nomi gave me a phone call saying she had 2 students working on red-absorbing visual pigments of long-winged butterflies. She said, "They need to know your methods. Come give a seminar." I did, and afterwards took them to the lab and showed them eyeshine of their butterflies. One of those students was Adriana Briscoe. After my visit, she contacted me with an unending stream of questions that continue to this day. Over time, that became a true collaboration, resulting in our first publication in 2011 with Richard White on the eyes of the painted lady butterfly Vanessa cardui. Many other collaborative publications followed, including a lot on butterfly species that could or could not see red. Gordon Pratt, yeah, these two are really special, those 2 guys. Gordon Pratt and John Emmel were always in the field, very knowledgeable biologists that evaluated diverse populations of butterflies all over Western America, especially those associated with buckwheat plants. They did a huge amount of work on Apodemia metalmarks. Gordon involved me in that work, asking me to measure eyes from isolated populations he regarded as subspecies. He told me about John rearing metal marks from eggs in his Hemet, California backyard. I met John, and in 1987, John started sending me Apidemia pupae for my work. That formed the keystone for what I'm about to show you. They published their extensive work on buckwheat metal marks in the magazine American Butterflies, all 28 pages of it. Here's the title page. Maybe or maybe not. What the heck? Now, why can't I? What?
Can you use your arrow keys?
Oh, there we go. I was hitting the wrong button. Okay, so this is the title page. And as you can see, the Mormon metal mark is a spectacular butterfly. See, where am I? Yeah, okay. So when younger, let's see, let's go to the next slide. When younger, John and his brother Tom Emmel made numerous contributions to knowledge of California butterfly ecology. John's day job was dermatology practice in Hemet, California. But noted here, John's free time was spent developing techniques for rearing various species of butterflies from eggs or collected larvae. John died in 2022. I dedicate this presentation to Dr. John Emmel. This is an example of butterfly eyeshine of the painted lady. And illumination here is from a 21-degree cone of white light. And the eye is on a curved surface. So each one of these units of the compound eye is looking in a particular direction and is probably a couple of degrees different from its neighbor. And the size of the visual field of one of these is the order of 2 degrees. So what causes eyeshine? It's a reflection from a white reflector. Well, let's see, what do we got going here? We have a lens that focuses light on like a fiber optic that contains the visual pigments that mediate vision. And it's like a waveguide that contains light. And at the bottom of that is this white reflector made by the tracheolar system that causes the reflection. It's white, but why is it colored? That's because of that waveguide containing the visual pigments. There's 3 different kinds, and they have peak absorbance and what we call the blue, the green, and where we can't see, the ultraviolet. Tim Goldsmith wrote this wonderful article in Scientific American. Tim was head of the biology department at Yale and was my mentor. He taught me so much. So anyhow, this is a really good article. How do we see colors? Most humans have 3 kinds of photoreceptors, cones, in the center of their retina with peak sensitivity at 3 different wavelengths. So our brain creates color vision by comparing neural outputs from those 3 types of cone. This is a complicated slide, so let's start. Well, first of all, 3-dimensional color vision of people. And so any color we can see can be created by a mixture of 3 colors. So let's focus on this slide here. What I've done here is taken a rainbow and made it not curved, but a bar. So think of this as, violet down to red. So this is our limits of our visual spectrum. And then the numbers here are called nanometers. It's just a way of referring to a particular monochromatic wavelength. And then on the vertical axis here, we show how absorptive a particular visual pigment is. There's a peak. Oh yeah, unfortunately, well, no. what about names? How do we name these things? Well, you can see there's several going on here. We name it for the wavelength at which sensitivity is greatest. Example here, this guy here peaks in the green. So it's called here pigment 530, and also it's got a label M. Sometimes it's called rhodopsin R530 because of the opsin protein that's part of the visual pigment, or simplified to just P530 or R530. So you'll see all of that stuff in this talk. Unfortunately, many publications, it's called a green receptor, since it peaks at a wavelength that we see as green. Nope, that's not it. It's not a receptor that signals green. Each one of these of itself is colorblind. That is why you're colorblind in really dim light when you're no longer seeing with your cones, but with your rod photoreceptors that all have the same R500 visual pigment. Some people have only two of those. They lost one of they never had one of the two long wavelength sensitive visual pigments. They're called dichromats because they're just two-dimensional vision. So what do they see? Well, to them, the color of the rainbow are gray, then yellow and blue. They can see color right there really well. But then out in the red green, it goes to gray. So to them, ripe cherries are the same color as leaves. Red and green traffic, that's a problem. Red and green traffic lights are just two shades of gray. Birds, well, butterflies too, later you'll see, they have another photoreceptor type that is sensitive in the ultraviolet. So, don't look at the M, look at the white here. Otherwise, your brain's going to think that that's sticking out of the page. Here, look here. Anyhow, so they have another dimension. So that color, oh, I forgot to tell you about the color triangle. Yeah, so if we think about, yeah, remember the S, M, and L. So this triangle has vertices that are each of the three receptor types. So if it were all nobody reporting except the S, you would be here. Same with the M, same with the L. And so this locus here is how these different relative amplitudes vary as a function of this monochromatic rainbow wavelength. And you see that all of these points are out in the middle someplace, except for down here with red, where we do manage to get down to L. So, and that's because all colors except red are have inputs from all two or three types. Red is peculiar, let's see. Yeah. Am I getting ahead of myself? Probably.
Very.
What would I do? Oh, yeah. Here. We're talking about UV and four-dimensional. So you see this here? That's really, it looks green to us. That's because we can't see that there's a UV reflection as well. So that's where we quit seeing and the birds can see very well. So it's really a color. It's not just green. It also, there it is here. See, that's the greeny one, but it's also got a UV component. In fact, all of those colors have some UV involved. Let's see what else. Oh, what? I thought I got rid of that. Anyhow. Yeah, so now all visual pigments absorb ultraviolet light, including all three of ours. So why can't we see in the ultraviolet? And the answer is that our lenses, the cornea and lens that's inside of your eye, absorb like all but about 1%. So there's not very much UV reaching your photoreceptors, and you're blind to UV. Except we're primates and we can't see UV. Almost all other animals can see UV. So we're deprived. Some butterflies have only 3 visual pigments. Examples are Vanessa, painted lady, and Adelpha limonitis. Most butterfly species have 4. How did this evolve? It evolved by duplication of the gene that makes the opsin protein for the visual pigment. Here are examples. The ruddy copper, Lysina rubidus, duplicated its blue absorbing or blue peak, yeah, peak in the blue opsin, and shifted the peak to, longer wavelength out there in the cyan. Longwing butterflies, Heliconius, this was really fun. They duplicated their UV absorbing visual pigment and shifted it to somewhat longer wavelength. And this gave them dichromatic color vision in the UV. So this is really important in mimicry complexes where they need to see each other. So they can do it using this private channel. The birds somehow haven't figured it out. So anyhow, that's a really cool story. And then Apodemia mormo, the metal mark, duplicated its green peaking opsin gene and shifted that all the way out to 605. And we don't understand how that happens, but that's another story. So what's red vision? I sort of mentioned this earlier. Well, all of our colors, like let's see, here's blue. That isn't just S, but it's all 3 are involved in what we see as blue. What's this, yellow? No. Oh yeah, there's green, and it involves 2. There's yellow. It's kind of a balance between M and L. And then orange has got some L, but also some green, M. And then by the time we get out here to what we call red, call that red, nobody's reporting except L. So this is when, that's what we see as red, is when only this receptor type is reporting. And then this is exponential decay. By the time we get to 700 for normal intensities that we see in life, we're blind. So we don't see anything out here. Okay. So butterflies, I guess I mentioned this early, that some can see out in the red and some can't. So ours peaks at 560 and we're what, 0.3% sensitive at 700. That's our limit. So yeah, I mentioned that. Black. Heliconids are like ours at 560, so they poop out at about 700 as well. Vanessa, Adelpha, they're at 530, like our green receptors, same as their green receptor, green peaking receptor, Longwing 662. And then this is kind of fun. The Viceroy, which is a mimic of monarchs, they look the same to the birds and to the humans, but not to each other. The Viceroy can't see beyond 642. So, and this is really short for the longest wavelength receptor. And then down to metal mark. How did I do that? Well, anyhow, let's see. Okay, so this is way out, 756. Why? What good is that? That seems to be strange. Yeah, so here's an example. This is Apodemia mormo, female from Ellensburg area, and it's foraging on rabbit brush. So what do they need to see? Well, females have to lay eggs on the right place of the right plant, but they first have to scan the landscape that's full of many plant species to find their host plant. Once located, they have to assess plant health and decide exactly where on the plant to deposit the eggs. And their special far red color vision helps them do that. I mentioned health of leaves. Here's an example of what's important and what happens with leaves. In this case, it's talking about nitrogen content. That's really important. and it shows, well, it's green. We can see this is green, but how green it is and what the peak is depends upon nitrogen. But then what's going on here? It's actually brighter out here in the far red than it is in the green. And the reason is chlorophyll. I guess there are 3 different kinds of chlorophyll molecules, and they all poop out at around 700 and don't absorb anymore. So they're actually much brighter in the far red than they are in the green. We can do the same thing and ask, well, how does this change with water content? Same story. So water, of course, is an important component of plants, and both of them influence the shape. And then if we look at Apodemia color vision, it's beautifully set up to evaluate color over that whole range, including the far red. Now, color vision works by a first order opponent interneuron. It's just a nerve cell that connects 2 different kinds of spectral types of photoreceptor. And then it looks at the difference. So you see here where one's going down, the other one's going up. In here, that difference is changing rapidly as a function of monochromatic wavelength. Same story over here in this part of the spectrum, this part of the spectrum. And then by the time we get down here, then it's just R605 involved with red and far red vision. So the color vision of this butterfly is beautifully set up for assessing plants.
So can we just stop there for a second, Gary, and just go back to that previous slide?
Sure.
Just to make sure I understand. So you have 4 curves there that represent pigment absorption of 4 different pigments. Is that right?
Yeah, 4 different visual pigments, right? Yes.
Okay, and humans have only 3. So this particular butterfly had some type of genetic mutation of one of the pigments and created another gene that could absorb light in another part of the spectrum. Is that right?
Yeah, that's what it took the gene for this one and pushed it out there somehow. Yep, exactly.
So
The duplication was here.
So it gave a lot more granularity to the color vision, and then also extended it into the far red wavelengths. Is that right?
Yep. Yep.
Okay, great.
Like the birds, and you need a color tetrahedron to describe the colors that it sees, maybe. Yep. You've got it.
Thank you.
How do they figure out what?
Oh, that's evolution. It's I'm not really qualified to say anything very erudite about exactly how that goes, but it's basically that as a species changes its habitat, its requirements, it is able to change the characteristics of its visual system to match what it needs. I guess I should say some erudite thing about that, and that's that the what evolution, the changes evolution has to make are very trivial. It's in that protein, which is barrel-shaped protein. It's got the little 11-cis retinal vitamin A thing that actually absorbs the light. Is all it has to do is change 1 or 2 amino acids in that protein, and it can change the spectrum. So it's pretty easy for evolution to change. It can happen on an evolutionary scale very rapidly.
And that enables them to have better fitness and better, they can reproduce more effectively. Is that right? Because they have more.
Well, yeah, they have a bunch of different requirements for vision. And
Gary, rather than
tweak.
changing an entire gene, is the change at the allele level? You just have to change an allele or 2? A portion of a gene?
Oh, I don't know, Bob. I can't answer that.
It's just a couple of amino acids that need to be that are mutated, is what he said. So I don't know what that means. I'm not a biologist either.
Yeah, in the sequence for the protein, it's just a matter of changing a couple of the residues, that's all. But the ones that are closest to the 11-cis retinal, because there's a difference in electrical charge that causes that. Okay, so back to the slide here. This is bean. I wanted to show you, you know what a bean leaf looks like. Yeah, there's green and there's bright. And then these are two host plants for Apidemia, and you see that they also are much brighter in the far red, although they look different in the visible to us as well as to the butterfly. And viewed by our red receptor and Apidemia's far red receptor, you see that when things beginning to change here in a big way to the reflectance of the leaves, we're not seeing it, but Apidemia certainly can. Okay, this is a video taken with me and Charles Remington at the Rocky Mountain Biological Station in Colorado in 1988. We were working on 4 species of coppers, and this buckwheat was Eriogonum subalpinum, was a host plant. So I was interested in what it looked like in the far red. And Eriogonum is the metal marks love Eriogonum. So this is also a host plant for Apidemia.
So
And
you're
the
just
leaf-
showing that it's very bright. Is that right, Gary?
Yeah, they're very bright. Well, yeah, look at how it compares to the background. Oh, particularly when you look at the flowers that are up on that stem. Let's see. But see, there's a leaf that's really bright in the red. That's an old leaf. The young leaf, there you go. See how bright that is? That's a big signal in a landscape that has 100 different species of plant. So it's really good at long range and close up.
Okay, can we take Melanie, do you have a question?
Is there a question?
I'm trying to find out.
Unmute.
Melanie? Melanie, can you unmute? Okay, let's go on then.
Sorry.
Okay. So
Um,
this,
So Gary, you said that Apidemia has better vision than other species. It's better adapted to.
no, I'm not making that claim.
Okay. Yeah,
We're not doing comparison here. We're just looking at what, Apidemia, why would it want to push that out to
I see.
and what good is it? That's what I'm focusing on.
So it sounds like, if I'm getting this right, is that the host plant for Apidemia is very, very bright beyond in the far infrared. So in a place that we can't really see it as bright, the butterfly can see it as bright, and that's its host.
You got
plant,
it.
what
Yeah,
can distinguish.
that's the main point. And
Does that answer your question, Melanie?
I think so. And also I was under the impression that that the Apidemia had detection ability, and that I'm glad to have that clarified. So it has specialized visual detection of what it needs to find its host plant. Is that what I'm understanding?
yeah, you got it, Melanie.
Okay, thank you.
Now, there's another thing. Once finding it, what are you going to do? Where are you going to put the egg? How, for Eriogonum, this one, the age of a leaf really affects the spectrum. Look at this. This is a young one. It looks sort of like bean, but then and this is the upper surface, and this is the lower surface. So the top and bottom are different. And then as they get old, here's an old leaf, and look what's happened to it. spectrum has changed a lot, particularly in the far red. And then the umbel, the flowers, this is what it looks like. So it's the color vision of the butterfly is set up to evaluate at close range where on the plant should I lay the egg. I don't know where that is, but they do. Oh, and this is the color vision is set up to see that. It's pretty obvious. Okay, another one is colors of wings. I just love this butterfly. This is a female foraging on rabbit brush, and she'll spend 20 minutes doing this. And it can often she won't even go to another bush. She'll just do it right on that bush. But she's constantly waving her wings. And as what I want you to notice is the shine of the wings if she gets the angle right, like right there. See it again. Yeah, you can see it on that wing at the start of the video. And then there it is, right there. Okay, so I mean, that doesn't really, it's not so bright to us, but it sure is to them. Let's see. Yeah. Here is my, sorry about this, Primos. This is really bad data, but it's all I had. I made an attempt at measuring this a long time ago. And this is different patches on the wing. And it's pretty clear that things are really bright in the far red. But as what I wanted to prove was that it's also bright in the UV. And my setup is really stinks when it comes to making UV measurements. So this was probably trying to go up, but it was showing some fluorescence that screwed it up. This one I didn't take far enough, but that was surely heading up here. So anyhow, it's bright both in the UV and in the far red, and I haven't proved that very well, but that's all I had. Okay, now, Gordon, yeah, 209. He, as I mentioned before, he's interested in populations of Apidemia at different places. He lived in Anza, and he had a lot in his backyard and that whole area. He had, well, yeah, something important I haven't said yet is that think about dispersal of butterflies. Apidemia doesn't do that. They're really homebodies. They hang around. And Gordon had different populations that he thought had been so isolated and localized and hadn't dispersed that they had become subspecies or possibly even species. So he wanted me to help him measure that. And he gave me this box. These are all Apidemia species. And then we have two more down here. And that looks like an Apidemia, but it's not. It's the Editha checker spot. But over here, we have more metal marks. These are from Arizona, different genus. And this one, little Alephilus, is lovely. Okay, so these, oh yeah, over here are buckwheat blues. And John and Gordon wrote a huge monograph about them. This is a particularly lovely butterfly that is loved by Washington Butterfly Association people, Sheridan's hairstreak. And then we got a snout butterfly, hackberry butterfly, red spotted purple, and pipevine swallowtail. And I'll show you something cool about its rear wings pretty soon. So I wanted to know, what does this look like in the far red? Here.
It looked like the silver satyr.
Isn't that amazing? I mean, and notice that it's not individual patches, it's the entire wing that is hot. So you can imagine this female sitting there flapping her wings constantly. That's a huge signal to the male if he wants to pay attention. Questions?
Well, I agree with Bob, man, that the silver, the brightness of it is just, it just takes your
Isn't that
breath away.
amazing? And we're blind to that, of course.
Yeah,
Little bit.
yeah. I mean, it really gives you an idea of what a butterfly might see by showing us that infrared imagery.
Gary, was that case of butterflies, that box, that side, was that sorted by gender at all? Did you get shine from both males and females?
Well, yeah, but I don't know. Did he have sex there? I have a separate list where he says exactly what they are, the sex, where they're from. So I have that detailed information.
You can tell that some of those are certainly females. Um,
Well,
Not all of them,
some
though.
of us can, some of us can't.
Well, the ones with the bigger, fuller wings,
Yeah.
and the pipeline swallowtail is less blue and greener than the male. That's female. Fuller winged on the red spotted purple and hackberry, certainly females. And then that one apidemia above the two big black and blue ones, that's a female. But there's some males there too. There's both sexes there.
No, they're all bright. That's
Interesting.
clear.
Any
so-
other questions, comments?
Well, I thought he was going to ask something about the swallowtail.
Oh, it's coming. Okay, so I've been working on polarization vision as well as color vision my entire career. Bees, flies, butterflies. Lately, I've been working on how butterfly eyes are designed to see patterns of both color and polarization when they're close to one another. So here are the butterflies on publication that set me on that research path. This is the Melissa blue. This is the Itis blue. They're very closely related. And note the pattern. This is a female. These are females. And notice the pattern on their wings. Oh, yeah. What's the courting game here? The females park and the males fly around looking for the female. And the signal the female is presenting is what you see here. They just sit there with their wings like that. Okay, so as far as visually and pattern, the difference between the two, they both have this circular with bars pattern, but here the black dots are fatter and here they're skinnier. And then here they have these orange bars that are wider than these yellow-orange bars over here on this one. So here's the work. This is Shapiro's lab, wonderful lab for years and years and years at UC Davis. And they did behavioral experiments to see what was important as far as parked female being attractive to the male. So they, they indicate their study showed that at least pattern elements are important for distinguishing the species. Those spots and the crescent-shaped orange bars. So they did photoshopping and presented these photoshopped patterns to males that were flying around looking for a courtship. And the results of their work said that these elements and the pattern are effective mate recognition signals. So here's what they did. This is what I showed you before. That's the real butterfly. These are photoshopped versions too. They did it. Well, there's a lot of these, but they did it sort of gradually reduced spots and saw gradually how the attractiveness changed. But anyhow, so here's an example where they reduced this. It's this one, except they reduced the size of the spots. And then this other, but kept the bars the same. And then this other one is where it kept the spots the same and changed the bars to be narrower. And yeah, both are important. It's the pattern. It's not just a particular feature. It's that pattern is important with those two different kinds of components. And in their discussion, they said there's probably other features of the wings that are also important, but they didn't know what they were. I have a suggestion. If this is Melissa, and if you get the angle right, remember this pattern? If you get the angle right, you can see that down here on the black area, there's really some decorations. And as what I've done here now is made photographs through a Polaroid sheet, where here I had it horizontal, here I had it vertical. And it doesn't look like much. In fact, it looks like that's better. But the deal here is that I've way overexposed this. This is much brighter with horizontal polarization than it is vertical. I have a video soon that I'll show you that makes this point much more convincing. So before I show you the fun stuff, any questions?
Gary, I'd just say that these are pretty subtle distinctions, even for taxonomists, such as Nabokov, who counted scales and looked very carefully at those very butterflies and their distinctions. So these butterflies are visually discriminating some pretty subtle pattern differences. It gives us pause. Yeah, it gives us pause to think about the whole question of acuity. When we were brought up to believe that insects could see shadow, light and dark, and here they are making pattern distinctions that would make any taxonomist proud.
Well, this is a problem with anthropomorphic science,
Yeah.
Yeah, it's a matter of scale. I mean, you can't think of showing a butterfly a Snellen acuity chart and ask them, what do you see? It's a matter of scale. When they get close to one another, like a body length or two, their vision is highly acute. They can see like 2 degrees or something. So if you break up a pattern of those butterflies at that range with that acuity, it's pretty darn good. And
Yeah,
this
and it's not pheromones
a
overriding
matter
it,
of scale.
because they were deceiving the right species to take the wrong mate. So it wasn't the pheromones. So it really is visual.
Let's see,
That's
I have
very
more
cool.
to say about what they see when they're close up. So
Yeah.
let's go on here. Yeah, so anyhow, I'm not going to make your eyes glaze over by discussing polarization vision, but is what I'd like to do. The fun stuff is I'd like to share some videos associated with my work that I think to be really beautiful and quite unusual. Here we go with the Python. Now, I have a daughter in North Carolina, and I was visiting, and her husband is a really good fly fisherman. So we went, he was trying to teach me fly fishing. And so we're at this park and near a stream, and I was doing this, and then I saw this pipevine just kind of walking around flapping its wings, and I grabbed my phone, and here's what I recorded. I mean, it's incredible. Look at that blue, whitish blue, bright, extensive reflection. I mean, what a signal flashing around. The thing is, we can see that, but we can't see the huge polarization signal that's also part of it. You want to see that again? Anybody?
Sure.
Yeah, okay. That's so cute. It's like it's clapping its hands.
And that, of course, is a male.
Yeah. So I suppose there's a reason for this. Oh, got it good. Okay, so
Mm.
let's see. So I have a really good friend and colleague, Nanfang Yu at Columbia University, and he loves butterflies, and he has two sons, and so he was rearing these pipe vines in his garage for his kids, and so he had a bunch of pupae, and he offered to send me some. I naturally took it, and here's one that he closed and is feeding, and what I did was photograph it through a rotating Polaroid sheet. I wanted to know how polarized that reflection was, because I figured it probably was. Okay, I've got the butterfly feeding now. Now, ooh, look at this. I'm rotating crossed, lined, crossed, aligned, crossed, lined. My goodness, it's a big effect. Let's see. So here I'm trying to align it so I see them equal and get as bright as I could. Cross line. So I stopped it here because I want to make a point about what we're looking at. It isn't just the polarized blue reflection that's going on here. Look at this as a pattern. We have stripes of the blue. We have white crescent things. We have, I don't know, C-shaped yellow things. So there's a pattern going on here. And the question I'm raising is, what is important here? Is it just the blue, or is the pattern itself important? And could it be important to the female, the pattern? I have more to say about that in a minute. Let's see. Oops, got to get out here, I guess. There we go. I did the same thing with a Melissa Blue, but not nearly as well. Horizontal, vertical, horizontal. I was trying to do too much at once. Camera and Polaroid. So that's vertical. Horizontal. So that's not good. But is what I can do is stop it. Let's see. Where's my horizontal? Ah. Horizontal, vertical. What I wanted to do, horizontal, vertical. I'm supposed to have the ability to pause it. What happened? It's just usually there. Well, anyhow, right at the start, it's bright and then it gets dim. Oh, there, bright, dim. Yeah. Higher angle. So vertical. Sorry about this. It's not that good.
Oh, that's quite all right. It seems like you've really demonstrated that butterflies have an additional component of eyesight, which is to be able to see polarized light. Jonathan, did you want to weigh in on that? Can
Well,
you?
I was just gonna say that that pipevine swallowtail, that's not a mating thing so much as like he's alerting everything in the neighborhood that I am poisonous. You don't want to have anything to do with me, and I'm making sure that you know that.
Not only through color, but through polarized light as well.
It's just that blue flashing. I don't think that they look that flashy in nature, because I've seen pipevine swallowtails in nature. That one was a beacon.
Yeah.
He must have been really fresh, huh, John?
Yeah, yeah, yeah.
Really gorgeous.
So this is extraordinary work that you've been doing on the
May I
polarization.
comment on what John said?
Oh,
Please.
yeah, please.
No question, they're highly aposematic, poisonous, and advertised, but it's always difficult to tell whether a visual cue is advertising to potential predators or to the same species and sex, or to other males or to the females. Very difficult to tell the difference there. I've watched a lot of pipevines, and they can, in the right light, be absolutely the males. The female is much duller. The male's extraordinarily flashy blue, mylar like a morpho. But that kind of very slow, flying near the ground, flashiness like that, rather than just ordinary flight, I don't know. That speaks to me probably of a sexual courtship signal rather than a aposematism. But it probably serves both purposes.
Yeah. Good.
Well, I agree. It serves both purposes, and that's great. When that works out. But I was looking at that as basically saying, "Don't eat me, don't eat me, don't eat me, don't eat me."
It's a big part of it, sure.
Surely a signal. So
Cool.
continue, Gary.
What?
Continue.
Okay, sure. All right. Yeah. So, my idea is that when they're really close, they can see at pretty high resolution. So what are they capable of seeing when they're this close and moving? And remember, this is slow-mo. I'll do it again so you can see how fast things are going at the start. That's real time right there. So this whole interaction is a second or two at most. So can they see anything? I have another example that's really fun. Maybe. Come on. Well, I got to get way out here, I guess. There we go. Okay. My daughter, Lucy, lives in Portland, is a Pacific Crest Trail through hiker. She hiked from Canada all the way to the Columbia River. And at the time, I was really interested in the Mariposa copper. And so Lucy, when she came across, well, I was her trail angel. So I'd meet her every week or less at a trailhead, but I'd get there 3 hours earlier, hike in, and then hike back with her. And whenever she came across any interesting butterflies, she would photo, measure the, note the GPS, and give it to me so I could go look at that population if I liked. And here's a video she took that's just striking. This is real time. It's a female being chased by a male. And you think, oh, yeah, well, if she's just rejecting him. No. If she wanted to reject him, she would have taken off immediately. She can fly anytime she likes. This lasted 30 seconds. And it isn't just her running and him chasing. She stops several times and looks at him. I think is what's happening here is do it again. She's evaluating his. And she can see polarized light. They both can. I can prove that from my work with their array. And so I think is what she's doing is I think that polarization is a real important cue because young males have a lot of nice scales, and then as they get old and banged up, they don't have so many. So just simple looking at overall polarization brightness, which should be a beautiful cue for fitness. So let's see, where does she turn? Right there. And later on here, she spends quite a bit of time relative there. looking.
But Gary, what's she evaluating here? Because if he's old and has lost a lot of scales, that might suggest that he has good genes for longevity. And that could be a fitness indicator. Or is she looking for freshness and lots of scales
Well,
and considering that to be the fitter male?
yeah, I don't know, but it's interesting because she spends a lot of time looking at him here. And then in the end, she says nope and flies off. But I don't know, this is just a fairly new idea, Bob, and I don't, yeah, it's more of an idea. I can't prove it, but I think polarization as a cue for fitness might be really important. But I'm doing comparative work and I've got a lot of things to say about that, but not here.
Very cool pictures in any case.
Thank you. Let's see, can I get out of here? Yeah, now, okay, so as I said, I'm doing a bunch of experiments trying to figure out how the eye is organized to see polarization as well as pattern and as well as movement detection for flight. Because the requirements for those two different kinds of polarization versus those things, the requirements are at odds. So what's the trade-off? I'm doing a lot on that. So I don't want to tell you about that. It's what I want to show you is the data I use to study that. Whoop. I just have to hit this, I guess. There we go. Okay. So here's some data. But I think it's just beautiful.
Can you describe what each image is, Gary?
Yep, I will at the end here.
Got it.
There. Okay, so what's going on here is this is eyeshine of the living intact butterfly. I'm illuminating with white polarized light, but I've got a polarized sheet that's crossed perpendicular. So is only the light that's coming through that's photographed is depolarized eyeshine. So these are photos taken 10 degrees apart. I'm rotating the whole eye. But as what I've done is Photoshop them so I derotate them. So I always have dorsal at the top. And this makes it much easier to compare what's happening with a given unit of the compound eye as a function of rotation angle. And as what's data for me is when one of these facets goes dark, because that tells me something about orientation of that unit of the compound eye compared to its neighbors. Let's see here. Whoops. If I can get outside of that, there we go. So here is the same thing from the same animal, but whereas that was in the part of the eye looking forward, here's a part of the eye that's looking up at the angle of 60 degrees. And I want you to note the massive color changes as the eye rotates. And if you like, you can focus on, say, that one. Let's see, here we go. Blue, white, pink, pink, red, red, white, blue, blue. Let's do it again. Let's see, let's do it again and focus on this guy. So what's going on here? Well, I think is what is going on here is that I'm looking, the color change, the reason for the color changes, I'm looking at different depths in the eye. It starts out being up where the part of the eye, the retina that's close to the lens is important, but when it goes dark, it allows you to see deeper into the eye. I just made that up. Okay. All right, here. I love this animal, brown elephant. It's so zappy. Uh, here's one pattern. It does the same kind of thing where these change, but this is really, I think, fun and possibly even important. What's going on here? It's the reason these are all lit at the same time is that their alignment is always the same. So this is like edge detection, but over this circular area compared to this one. And then inside is polarization stuff. So I think this might be a feature detector. But the reason I'm showing it to you is it's really cute. This butterfly is from Arizona. It's Palmer's meadowlark. It's really small. That's my fingernail. And you'd think, well, it's got these really tiny eyes, so it must be really deprived, but it isn't. It's got tetrachromatic color vision, huge acute zone in the front of its eye. And it's the first butterfly that I discovered this possibility of having color changes with that rotation. It's just really pretty. I'll just let it run. This is optical art. Waveguide modes. More waveguide modes. Okay, an hour's up, isn't it? Okay. Let's see, I'm getting close. Let me show you this. This is really important for the far red thing, and also Gordon Pratt. This is the golden hair streak, male, female. It has far red rhodopsin. This is about as far red sensitive as any hair streak I've measured. Gordon Pratt sent me this, and he told me that they prefer being in the interior of an oak and that their courting and mating behavior happens at dusk or after on branches inside of the tree. However, for them, it's not dark in there because they can see with downwelling far red light not absorbed by chlorophyll. And I'm guessing that red sunsets may help a lot. The structure of the eyes is designed for that habitat. Usually butterflies have only one acute zone facing forward, but this species also has one facing upward that has photoreceptors loaded with far red rhodopsin. And also it's very unusual to have a lot of far red rhodopsin in the upper part of the eye. Okay, questions? I'm not
Have
quite
you
finished, but almost.
Have you ever done any work on the Stella orange tip?
Oh, yeah, I've done work on orange tips and I hate those things because they have no eyeshine.
Uh-huh. I was wondering about them. You
Yeah.
know, I go out in the field in Yellowstone and do all the butterflies there.
Uh-huh.
And I don't find the males in the same location as the females. I keep wondering, when do they ever
That's
hook
not
up?
going to work.
I know. I don't understand it. Can't help me out on that, huh?
No, they don't have eyeshine. I haven't been able, and that's true of the ones in Switzerland as well as the ones here. Yeah, the marbles, same thing.
Uh-huh.
I think, but it's fair to say that some orange tips actually do have eyeshine, like the Hebomoya. They have
Oh,
it. I
those
think
are the
only
big ones. That's no fair. I'm
the
talking
Antoharis
about
genus is the one without. And I think that also I think we should let the people here know that unfortunately also all the Papilio the Papilionidae are without the eyeshine.
Yeah,
So all the
thank you,
swallowtails
Kilo.
and all the big butterflies, they are also without it. So, right?
So why eyeshine?
So
Well,
Gary, can you wrap up and then we'll get to
Yeah,
more in-depth
definitely.
discussions because you're really close. So
Yeah, this won't take long, right? I was a member of the Yale University Department of Ophthalmology for 21 years, and I gave lectures to residents on physiological optics of human and insect eyes. And a very common question was, insects are so tiny, how can they see anything? I have an answer. In 2023, I gave a talk at the Conference on Invertebrate Vision in Sweden. They have a wild, fun crayfish aquavit party on the third night. Weeks before the conference, one of the organizers sent out a request for us to write a song that would be printed in a songbook so that all participants could sing after having sufficient aquavit. Here is my song. I'm not going to sing it, but let me recite it. This eye is your eye. This eye is my eye. We can see colors of the rainbow. We can see blue light. We can see green light. Your eyes are really not so good. We can see red light. We can see purple. We can see cyan. We can see yellow. We think we're so good, but we're deprived. Your eyes are really not so good. We see in dim light, but not in color. We can see slow stuff, but can't see fast stuff. We can see forward, but not behind us. Your eyes are really not so good. We can't see UV. We can't see far red. We can't see IR, can't see in starlight. We can't see any polarization. Your eyes are really not so good. Lepidoptera are so much better. They see UV, they see far red, they see IR, they see fast stuff. Your eyes are really not so good. They see behind them, they see in starlight, and they can see polarization. So be humble when writing papers. Your eyes are really not so good.
bravo, bravo.
Paulette, thank you so much for helping me to organize this thing. You were really, really helpful.
It was such a pleasure to work together, Gary. So now is our time for free and open discussion. And Jonathan, did you want to, you had a couple things I think you wanted to say.
did I want to talk? Did I want to talk? You have to ask that question.
Come on, John.
No, first off, I've known Gary for some time, and he's always very, very stimulating, and tonight's no exception. But I want to get to the one thing about Apodema mormo and their potential food plants. All of the food plants we recorded are basically senescent at the time that mormo flies. So they're going to be looking at old, beat up buckwheats. That might explain why they're so sensitive to older leaves, right? Maybe. All right. I don't know, but that's
yeah.
Yeah.
Oh,
Okay.
wow. That's cool.
Well, I thought it might be, but okay. So the next thing I wanted to say, it relates to a presentation I gave that you watched earlier. I don't know when, in the last 5 years about living wings, right? And we were talking about what wings are all about, in terms of how they they have a nervous system and they have a circulation system, and I concentrated on pheromones and the things that are pertinent to mating systems. And you came in with something that turned out to be very, very important. They also have sensors. And it turns out that these sensors allow the thoracic ganglia to control wing movement and negotiate whatever. Well, that's cool, except if they're not guided. I mean, anyone who's missed at a woodland skipper, and because they saw you coming, knows that somehow what they were seeing was communicated to the thoracic ganglia that evoked that response. And so the connection between those two things is really important, but the independence of them is also important, right? So like that was like totally cool. Now tonight I realized that I was thinking you were going to say infrared, and you eventually did, show that infrared's important for mate recognition too. But it's so much more important than that. I was like really astounded and enthusiastic, and I probably won't be right for the next day or so.
Got John, you haven't been
Thank
right for
you.
50 years.
That's
Well,
great.
I wasn't going to say that.
Well, what's that?
Never mind. It wasn't kind. It
Well,
was
that was a great piece of work, really showing that they have recognition not only of potential mates, but also food sources and laying sources. You know, where distinguishing host plants and their fitness. I mean, it's just astonishing that their mutations have enabled them to see in a range where they can distinguish those things.
Bob, you were making a point?
two things, Gary. One is that I was in Charles Remington's lab at Yale
Yeah.
when you first showed up there. And I just want to say that I remember to this day how excited Charles was that you were coming into this because he was interested in this area, but he did not have the expertise to work with it. But with Tim Goldsmith and Bill Miller and the others, he knew there was a potential team there. But you were the cog that came in that made it work. And then you made it work for what, 21 years there? So I mean, that was so exciting.
Yeah, 21 years at Yale, yeah.
Yeah. And watching Charles's ignition by your coming into the lab about at the very same time when Nomi Pierce was coming into his lab every week as an undergraduate,
Yeah,
getting excited at the very same time.
yep.
And she went on to her brilliant career at Harvard. But that was to see the ignition between two scientists who get together and they say, oh yeah, we could do something here. That's very, very exciting. I like to think that John and I had some of that feeling 50 years ago when we went to Bear Canyon together, although I'm not making comparisons. But you've done fabulous work. And it was really exciting to be on the ground floor of that just as a witness to see how you energized that entire department, as I'm sure you did at MIT before that and elsewhere. Second thing is a practical down-to-earth butterfly thing. I was really interested that your last thing you said about Haberdae's grunus, the golden hair streak,
Yeah.
because John and I were looking for that a long time ago on several failed expeditions, including one in the snow, as I recall, before we finally found it up there when Art Krukeberg finally told us where the chinkapin trees were by the big lava bed, and we finally found it. But we've known ever since then, but we read it. We read it in Comstock, we read it in Wright, we read it in Tilden. Everybody who knew it down in California talked about the crepuscular proclivities of this butterfly.
Yeah.
And we've confirmed that to some extent. I've certainly seen them and watched them flying at dusk and prior to that. But we never knew why. Why did they do that? And you have now given us a molecular clue as to a particular modality they have, so they can do something at dusk that cannot necessarily be done. And the red sky impulse. So just think how these plus chinkapin is fire adapted. So just think that the whole red glow fire sky thing might work in the long term.
Well, I was thinking also it might keep them away from predators. I mean,
well, that too,
give 'em
yeah.
this-
Well, exactly, because the bats aren't out yet, but the birds have gone to bed. It's a really good time to behave. It's warm enough that you can do that. So that's brilliant. I want to congratulate you on that. Thanks for a great talk.
Bob, I have another example like that. You know, Rhyodina lysipus?
Yeah.
Yeah. Well, Nanfang was down in Ecuador and he brought some back, and I had the opportunity to measure 3 of them. And they have huge amount of far red rhodopsin in their eye. So they're in the understory, and it's the same story, I think. They're using far red that permeates through the lack of chlorophyll absorption for vision.
Yeah.
I mean, wow.
Yeah, wow. That's cool. Thank you.
Yeah.
So David Berger, if you're still on, can you unmute? If you're not, then I'll go ahead and ask your question, or if you're having trouble. David, are you there? Okay, I'll read David's question. He says, you mentioned near and distance vision on the part of butterflies. How far can they see?
Well, good question. And the thing is, what is important visually for them to see at distance? The flashes, that's easily detected if it's bright, which it is. I've demonstrated that. So they are not seeing form, but color, polarization, and flicker. Flicker is so important for them. And they have eyes that are so fast that probably the long range cue is more, interspecific cue has to do with motion, flicker, and looking for, particularly in conspecific, if you know what frequency is typical for wing flapping frequencies, you can tune your sensory system to filter out for just that frequency. Things like that. So yeah, and then when you get closer, then more features can be important. But to get really what you would call imaging, you have to get really close. Otherwise, it's pretty much long range vision. And like territoriality, yeah. Bob, Charles and Ellen were out in the field while I was in the lab working on those coppers.
Right.
And Charles would, well, they would, there'd be a territorial male, and he would trapline, go round and around. And so he would stake out a territory. And then conspecific would come in, they'd go after him. So Charles and Ellen made notes about, well, how far away before he launches himself?
Ah,
And it's like 3 meters,
3
something like
meters.
that.
Ah,
3 meters, really?
So
Yeah,
that's, wow.
Oh,
So extreme acuity up close,
right.
but
yeah,
also 3 meters away.
Yeah.
That's
That's
very
pretty good.
impressive.
Okay, okay.
Yeah. Well, that seems to be, that figures for when a Lorquin's admiral will fly out at something that's 2 or 3 meters away. Same
Uh-huh.
with morning cloak or a tiger swallowtail.
Yeah.
Hey, Gary, do you know offhand the numbers of the flicker speed of any butterfly versus that of humans? The
Oh,
flicker.
Galmetus evagoras. The flicker. Oh, yes. There's been a lot of work on that.
Yeah.
Physiology of the photoreceptor. 200 hertz, no problem. Compared to what, 30 for us or 60?
There you go.
Yeah.
There you
God.
go.
Oh, they have really, really fast eyes, and they're, yeah, their spatial vision requires flicker. Absolutely, because the eye itself, the photoreceptors, there's probably 5 different types. They're all heterogeneous, so they have to scan.
Yeah.
So a big unknown here that I'd love to have somebody work on is, what do they see? How do they extract features? How do they use that flicker in their brain, stuff like that. There's so much we don't know. So much.
Hey, but so much we do. I went to a big seminar from people all over the UK on insect vision in Cambridge.
Uh-huh.
And I think that would have been 1972. It was like going to a seminar on AI in the Neolithic or something compared to what you know now. And a great deal of it is thanks to your work.
Oh, wow. Thank you.
So now's the time when anybody can unmute, just talk freely. So I do have a question. You were talking about eyeshine and about how some species don't have eyeshine. Is it possible to talk about that? I mean, is it the structure of the eye? Is it something about the receptors or?
Yeah, Bill Miller was the electron microscopist at Yale where we first discovered the tapetum. And very early on, we know that the swallowtails didn't have it. So Bill wanted to know, well, come on, they have tracheolar system, they have to breathe. So why don't they have a tapetum? And he did electron microscopy and showed that the place where in the eye where all other butterflies that have eyeshine do their thing with making layers, with swallowtails, it's just a glob of chitin. And the tracheos are still doing their thing of supplying oxygen to the retina, but it's just a bunch of pigment there that's absorbing the light.
Hmm.
Same story with the orange tip, Anthocharis for some reason. Why?
What is it, the entire PRD?
I've looked at a lot, including yeah,
Primosh, do you
yeah,
know if there's more than Anthocharis?
including
No,
I've
Primo should have Maya reflected, so there's one pyre that
Oh,
reflects
that's right, yeah.
eyeshine.
Yeah.
who?
They have, I think that most of them actually, so apart from the Antoharis, all of the pierrids that I've looked in had some eyeshine,
Hmm.
but very often it was very, very red, and that's actually for the reason that is additional to the ones that Gary has mentioned, namely, they have a little bit, I mean, just like it's interesting, like Papiliones, they also have a two-tiered retina. But they do have the tapetum, at least I mean all over the eye, apart from the orange tip. But they have apart from the dorsal part of the eye and the ventral part of the eye, they can have a lot of red screening pigments that
Wow.
actually sometimes shifts their eye shine so far that with our shitty eyes we don't really see it through the microscope.
Really?
Um,
I didn't know that.
Wow.
That's
so
great.
there was actually a thing, the way how, so we knew that, so from the work from Willy Ribby, who actually also did very nice anatomy back in the 70s and the 80s, right?
The
We knew
original.
that Paris Rape would have a tapetum and also an eyeshine. And then when Kentaro Arikawa, another, I think, quite well-known lepidopterologist, brought Coleus from Japan to the Netherlands, we thought, oh yes, that's just another Antoharis, because it seemed as if it really didn't have much eyeshine. But then with the
Yeah, no.
monochromatic CCD camera, we saw it,
Yeah.
and that actually is the reason why maybe we should also say, Gary, that there's another way of actually how you can start, how you can make yourself see red if you're a butterfly, which is to still have your shitty green rhodopsin, but to put very deep red sunglasses on, right? And actually,
Yeah.
this is maybe also like one of these like
Oh,
philosophical questions that can come up. Like, why did the metal marks seem to be the only butterflies that have really made like that red rhodopsin, right? Red peaking rhodopsin or whatever, like the
yeah.
600. And nobody else apparently for no other groups have managed to do that. And they have chosen instead to put the sunglasses on. Why?
Yeah.
Gary, I think that Primosh has just supplied a name for your song. The name is Our Shitty Eyes.
Excuse my French.
It makes the point. It makes the point, though.
So is that, I mean, I'm just trying to understand how that would arise. It must be some type of, mutation somewhere along the line. I was wondering if anybody's looked into, what came first, the glob or the eyeshine or from the tubes or the glob.
I mean,
Well,
Gary, please go ahead. I think
yeah,
you're the
yeah.
one
The
who
the
has seen
real
a lot of
function of
eyeshine.
what's going on down there is you've got a big tube, a tracheal, big single tracheal that's coming up into each of the units of the compound eye. And then it turns into a manifold. Its job is to split into, I don't know, 8 tubes that go up along and diffuse air to the photoreceptors at a really high metabolic rate. So is what's happened is that, tracheals have ripples in their surface on the inside. So increase the surface area so you get good transfer. And so is what's happened is the evolution is those ripples have gotten really wide, and they kind of come together, and it's really specialized those ripples that have formed the platelets of the tapetum. Now, why some did it and some didn't, but all of them have a tracheolar distributor. They have to because of the high metabolic rate of those photoreceptors.
Okay,
I
thank
know, I
you.
just made that up.
I got a question. Do we have any skippers with eyeshine?
Well, all skippers have eyeshine, but the problem is the optical source of their eyeshine isn't the same.
Uh-huh.
It's really bad. I mean, is what's happened is there's specialization of the tracheolar cells that surround the photoreceptors. And so nothing really goes through. But Primoz is really good. He managed to get eyeshine out of a skipper, right, Primoz?
Yeah, well, skippers are they have it's weird, but they have like the eyes is of that type which is normally associated with nocturnal Lepidoptera,
Yes. Yeah.
even though they are very much diurnal, all of them, as much as I know. So yeah,
It's specialized for diurnal.
so and they, as you may know, that I think that most of them are really fast flyers, right?
Oh, yeah.
So actually, for some reason, they have retained the same sort of eye as supposedly, well, I don't know where the whole phylogeny is going, but the talk, at least the original one, which is probably somewhere there, as you know, all insects should have just UV B and green peaking receptors, was that also all Lepidoptera, or like the ancestral Lepidoptera, would all be nocturnal and would have this so-called superposition eye. I'm not going into that because this may change. I don't know, but the fact is that within the butterflies proper, it is the skippers that actually have the nocturnal type of the eye.
Mhm. Yeah. Right.
And then there is also this weird small group called Hedilidae, which I think
Yeah.
are also in Chile, right?
Yeah.
They have also a weird eye that has one receptor cell less than the others.
Uh-huh.
So there's a paper from some Canadian group, I think, that actually with Eric Warrant, I think I forgot, but they studied that one Hedilida that apparently has one cell less, but also maybe but I think a diurnal eye. Forgot already. Yeah, it's weird. But yeah, skippers have a nice blue eye shine that looks not unlike the one that you can see in, let's say, in the macroglossum, you know,
Diurnal sphingids. Yeah.
yeah,
Same eye.
yeah.
Same eye.
So
This is
Alright.
kind of exciting. I'm sorry, but this is kind of exciting. I really like this. Is probably phylogenetic, but what that means, Hidalids. When you mentioned them, I was going to ask if you had done them. So, like, to tell me that you have, and they have the same kind of structure, maybe even more primitive than the skippers. The Hidalid.
look, I'm really not into the phylogeny of
Yeah,
this, but
yeah,
they
yeah.
were supposed to be like like they are less they are actually before the skippers, right? If I'm not mistaken.
Nobody knows, like,
Yeah, you see.
yeah, 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 yeah.
By the way, it's not unprecedented for one particular group of organisms to be separate from most of the others in a trait like that, like swallowtails not having the tapetum or tapetum. For example, if you look at tapetums in mammals, we're very familiar with them in cats and dogs and coyotes and deer. But I think most mammals, I don't know if it's most or many mammals, do have a tapetum and eyeshine. Primates do not, which is a problem for those who think that Bigfoot has red eyeshine. It would be just that would be
Good one,
that would be
Bob.
unique
That's
among
for
the
you,
mammals.
Bob.
What's that?
That's for you, Bob. You wrote the
No,
book
no.
on pictures.
Well, I said that on a 3-hour podcast the other night. When it started getting really silly that the red eyeshine, no, they don't seem to have red eyeshine if they exist. But that would be unprecedented among the primates. But so here you've got a big group, an order of primates that are different from their confederacy of mammals in lacking this particular trait. So for swallowtails to be that way too, Yeah, it's weird, but it's not unprecedented for a group to have quite a different configuration in a given trait from its confreres.
Good point.
Phew,
Okay.
love the discussion.
Any other questions, comments? Anybody want to bring any other suggestions up for butterflies that might have been looked at? Okay, well, this has been a really wonderful talk, Gary. It's just
you.
really marvelous, really exciting to hear, in our volunteer group about cutting edge research that's being done currently. So we are just really thrilled to have you join us and talk about this. John?
No, I just give him the peace sign. That's
Okay.
wonderful,
Uh,
wonderful.
Could I say one last thing?
Yes, please.
I just want to say how wonderful it has been for me to associate with the Washington Butterfly Association and the local expertise and the free way you share what you know. And, you are so stimulating to young people that want to learn, and the field trips and everything. It's been just wonderful for me and so helpful in my work. Thank you, guys.
It's a great partnership, really and truly. Thank you. Yeah. Okay, so thank you, Gary. Just marvelous talk. Really enjoyed it. I just want to mention that next month our talk will be by David Droppers, and he'll be talking about his magnificent survey in the North Cascades.
Hey, Paulette, do you want to tell people, give them an advance notice of Stu's talk he's going to do too?
Yes, I will do that. So April, we're going to have Julie O'Donnell, and she's going to be talking about pollinator gardening. And so she's been really branching out. So she's not only looking at butterflies and moths, but she's been looking at bees. So I think it will be a very, very interesting talk. And we're going to see if we can entice some of the bee people to come and join us too. And then the following month, that would be May, Bob has kindly contacted one of his colleagues who has been doing some reintroduction work in the Palisades in California. Why don't you talk about it, Bob? Because you've worked a lot on that.
Oh, no, not the Palisades. Ooh, ow.
Oh,
No,
I'm sorry.
not the Palisades of Presidio.
I'm sorry,
Oh, the
Presidio.
Presidio.
Yes,
Oh, okay.
not New
Wouldn't
York,
work,
California.
wouldn't work out too well the Palisades right now.
Good work, Brenda.
A good many years ago, 20 years ago or so, I wrote an essay called Resurrection Ecology. And I'd been influenced by Jeremy Thomas and the work to bring back the English large blue, which had become extinct after a long history of attempted conservation work. By getting the habitat back in shape, once I realized how and what it needed, and then bringing back the Swedish large blue, which is about 10,000 years or so removed evolutionarily.
Wow.
And it's taken very, very well, and it's eating the ants out of house and home. It's doing well. So I thought, well, Xerces blue is gone, but now that the Presidio, where it was last seen, has been brought back to
Whoa.
public ownership, and a lot of the habitat's been restored to the San Francisco dune habitat with many of the plants and animals, 3 butterfly introductions already, but including the food plant of the Xerces blue. What harm would there be in bringing in the nearest genetic relative in the Glaucopsyche group, the nearest subspecies of Glaucopsyche ligdamus, the nearest silvery blue, and plunking it down there and seeing what happened over time? Couldn't do any harm, and it would be very
Yeah.
interesting to see if microevolution began to bring back something with Xerces-like traits. So I
Wow.
published that. It was reprinted in a few places and a lot of hemming and hawing. And then about 10 years ago, some people took interest and decided to try to do it. 5 years ago, they really decided to do it. They got permits, they got looking, and Stuart Weiss headed up the team to determine the genes of it. So with the genomics team at the Cal Academy, they went out and they found out which subspecies of the silvery blue, it's one near the Marina Dunes, as it turns out, I don't remember his name, was the closest to what Xerxes would have been once they ran its DNA. And so that has now been done. And
Cool.
we're a couple seasons in. So Stuart Weiss has been a major figure, former student and colleague of Paul Ehrlich's at Stanford, major figure in conservation of checker spots and the conservation and ecology and biology of checker spots in the Bay Area and of monarchs. He's done a lot of the important work on individual monarch site management in California. But he's been the one person who has kind of brought all the interests together to do this re-intro at the
Wow.
Presidio. And he's going to come and
Cool.
give a talk in May. That'll be fun.
Great.
Yeah, I think it's really impressive
great-
how much
great
work
guy.
they did for habitat reconstruction before bringing in the butterfly. I mean, it's a long story, and I wanted to find someone who could tell that whole story, like from the beginning, because it was such a long lead time to get
Yeah,
to where we are now.
it won't work unless you do that. And that's what Jeremy and company did in England with Maculini Orion Eudifron. And that's why it worked. So yeah, Stuart's a great guy, very charismatic. Gary, I think he's going to have a hard act to follow in you and everybody else. But it's going to be a great talk.
Oh, thank you, Bob.
Thank you. Okay, well, we hope to see everybody next month, and we want to welcome especially our guests who are joining us from afar, and it's been a real pleasure to meet some new folks and have some fabulous discussions. So thank you very much. Thank you, Gary.
Thank
It
you,
was just
Paulette.
marvelous. Okay, we'll see you next month then.
Okay.
Oh, I did want to mention too that Primoz put some references in the chat, and I think I'll be able to get a copy of that. So if anybody needs it, just contact me and I'll send it to you.
Thank you.
Okay.
Thank you,
Good night,
Primoz.
everybody. All right. Take care. Good night.
Okay.
Cheers.
Bye.
Bye.